Hi, Professor Harrell. Kendall here. I'm going to go over my first lab, and I'm going to show my results using the tryptic soy broth tubes, just because I think they're going to show the easiest on here, and then I'll show my mixed media culture plate, just because it's great results on that. So I don't have my little jars, or else I would show you. I think I mentioned this in my lab report, but my husband threw out the small bag after I already did my lab, but they were in a pile with other trash and thrown away. So I do not have that, but I do have the results, which is great. So they're over here. Oh, my labs. So this is the E. coli. There's some sediment at the bottom, you can see, but it's pretty clear. You can see through it. So that's that one. This one is the Micrococcus luteus, and this is even clearer, I think. But there's a little bit in the corner up there at the bottom. This one is the staph. This has same thing, very clear, a little bit. There wasn't too much, which I honestly thought I did some of this wrong because of that, but I don't know. And then the last one, the serratia, is really nasty. So it's very cloudy. There's a lot on top kind of floating at the jar. I think you can see like right there. And then on the bottom, there's just a ton of stuff, and it's really gross, so that was fun. But I think it went well. My house is clean, but I do live with a dog, so I think some of like the contamination of just like the dust in the air could have contaminated it. Truthfully, I did not wear a mask. I did say that in my lab result. I wasn't wearing a mask when I did this, and I work with a lot of patients and everything, so I guess I should have thought of that and worn a mask when I was doing it. I did wear eye protective wear, but yeah, so I think that it might have caused some contamination or something. I was very vigilant with the flame. The flame though, the little topper, it didn't really work. It kept going out. So that was also another cause of possible contamination. I had to keep relighting it in order to flame it, which was frustrating. But I think during those moments where I had to put the lid back on and restart it and flame it again, I think just the time could have allowed for more contamination. So it might have not been the best results, or I probably would have gotten different results if I was in the lab at the college. But I think it did pretty okay for what I had. Show you results. Yes. So I showed you those. I think this was the second lab. I know I'm a little behind. But we had to do the mixed culture, and I got that, and I just wanted to show you this because it was pretty cool and it was kind of grody. But this is the mixed culture, and it's just wild. Yeah, that was pretty wacky. Sorry, I'm trying to get the best angle, but yeah, so you can see all the colors and stuff. But that was pretty cool. I don't even think I got one like that when I was in your class before, but that's wild. So the process, I have my notes up here. So I reactivated the the bacteria and got that going with the broth, had it sit for like 30 seconds and everything. And then I took the sterile pipette and took it out, flamed the loop and everything, or not the loop, but because we had plastic loops, but flamed the test tube and got some bacteria, put that in, flamed it again, turned it off, not off, but I put the cap back on with my pinky, put it back over here, and threw away the pipette and the loop, and now my jars that had the bacteria in it. So that's what we did with that, and then had to do the same thing with the slant. Lots of maneuvering with things in hands and using the test tube rack to being able to and grab the bacteria and then take it out and then put in the other one while capping it. Flaming the mouth of the tube was super important. When I did the plates, I did what we did in class last time, and so when I got the bacteria from the loop, I would open it and do the pattern down, and then turn it, do another pattern, turn it one more time, and go up. And then I put that back on and threw away the loop, discarded that. So that was kind of what I did with the 3 processes. But after I finished with the soy broth, the tryptic soy broth, I put it, there's a small table in our living room, and I put it over there because we have a large window, and I left that open while I wasn't here during the day for the sun to catch it, which is why I actually think that this got so grody, because it was in the spot where most of the sun hits. But I left my tubes there for 48 hours. Not much changed. I truly didn't notice anything in the first 3 until we got to the serratia, and then it started getting cloudy at first, and then the ring on top. After 72 and like 96 hours, they all started to look how they are now and stuff. And honestly, the serratia, it probably even got more of this white dotted grossness, bacteria stuff on the sides of the test tube, and that happened actually at 96 hours. So that was kind of cool. So I think everything kind of stopped around 4 days, and they've just been sitting to the side for now. So yeah, that was kind of the first lab experience, and I'll catch up with you in the next one. Okay.